The American Brain Tumor Association is pleased to welcome you back to our webinar series. Our webinar today will discuss molecular testing, how it's improving brain tumor diagnosis. My name is Alex Sierra. I'm the Research Grants Manager here at the ABTA. I'm delighted to introduce our speaker today, Dr. Keith Ligon, MD, PhD. Dr. Ligon is an assistant professor of pathology at Harvard Medical School and the Dana-Farber Brigham and Women's Hospital, where he serves as Vice Director of Neuropathology. He's a board certified in neuropathology and a principal investigator in the Dana-Farber Cancer Institute Center for Molecular Oncologic Pathology, where his laboratory performs research on adult and pediatric brain tumors. Thank you for joining us, Dr. Ligon. You may now begin your presentation.
Thank you very much, and I want to thank everyone for joining us today. I am going to speak today about molecular testing and the exciting changes and developments that have happened over the years that have really worked towards improving brain tumor diagnosis. And this is going to be a complimentary talk where Dr. Patrick Nguyen, I believe, is going to give a talk on the implications of this testing for the therapies and things like that later on from the ABTA. And I do want to encourage right up front that if people have any questions that they would like to reach out, certainly feel free to email or call after the webinar if you have any questions or require any other clarifications of anything that I've said. And I just want to point out that I'm a neuropathologist who practices both in adult and pediatric brain tumors. And so all of the concepts that we're going to talk about today would apply to adult or pediatrics. I'll highlight some of each test and how they might apply to each. But really the testing and the approach is going to be very applicable to any type of brain tumor patient. So I think the first thing that I'd like to discuss is how do pathologists really diagnose or work to generate information about brain tumors for patients, and how is that information used? And this is a process which pathologists undergo every day. We really work hard to try to generate diagnoses that are reliable, that don't change from place to place and opinion to opinion, but it is a very organic process in a way, and involves multiple steps of information, data, and impressions from both pathology and treating physician teams. And so what I've diagrammed here is kind of the thought process of what a pathologist goes through in a first approaching a brain tumor sample or patient's diagnosis. And the first step is going to be to assign the tumor a tumor class. And this is really what we think about as being the actual diagnosis of a brain tumor. So something like a term like glioblastoma is a diagnosis that a patient is given. Each different tumor class is given a tumor grade whenever possible. Sometimes it's not possible to assign a grade, but the second step is the grade. And so you really need to know the diagnosis in order to accurately decide what type of grade the tumor would have. And what do we mean by grade? Well, the grade is somewhat related to the biology of the tumor as to how we expect it to behave. And what it looks like under the microscope, as far as does it look like a better or worse variant of that particular type of tumor. And we'll talk a little bit about how we might do that with molecular tests later on. And each of these assignments of class and tumor grade and categories have strong implications and impact, and basically determine the treatment that an oncologist or radiation oncologist or surgeon would expect their treatment, what they would want to apply to a patient. It really is dictated to a large degree by this process. The response that's expected to that treatment is also determined by what we know about the tumor for class and grade. And then ultimately the prognosis, or how we expect a patient to do in the long run, is also determined by these types of information. And so what I've listed below are the two ways in which pathologists generate data right now routinely in order to make these predictions and help patients and their treatment team with decision making. And the first and most important still in today is what we call H&E or histopathology. And this is really the cornerstone of diagnosis for all types of pathology still. It involves a microscope, and the microscope is a tool, if you think about it, that we've been using since the 1600s in order to sort of look at tumors and diagnose patients and know more about them. So it's a very old technology. There's a lot of concern or discussion and debate about whether molecular testing can replace H&E and histopathology, and in some instances that might be a relevant discussion, but for the most part it's a very efficient tool when you combine a microscope with a pathologist with sort of knowledge of what we have from previous days and ages. You come to very good conclusions about it. And we have consensus statements and rules that we follow in order to diagnose tumors. But what we're going to really focus on today is how that's improved greatly by adding molecular testing into the mix. And there are several tools we'll go through and discuss that are used under the heading of molecular testing. One is Immunohistochemistry IHC, which is a protein test. Cytogenetics, which is a DNA-based test, looking at generally the number of copies of genes or chromosomes. And these tests you may have heard of are FISH or Array CGH. We'll talk about sequencing technologies, which are one of the most recent types of technologies to be applied to patient and brain tumor diagnosis. This is also generally based on DNA, and you may have heard terms about exome sequencing or whole genome sequencing, which are coming into practice for brain tumor patients. And the last category, which is not used quite as much, but you may have heard of as well, is expression profiling. This is generally based on RNA analysis. And like I said, we won't go into great detail on that because it's not used quite as much for clinical care at this point. So how do we generate or obtain all this data? Well, the process of generation of the data really requires an integrated approach to the diagnosis and also involved in research. And what I've diagrammed here is an example of the type of process that goes on behind the scenes for the most part in order to generate all kinds of information and data for patient care. The first step is usually very critical, which is a consenting of patients and discussions with patients about what might happen to their sample after it's been removed, if there's a biopsy or resection done. And this step really is important for brain tumor patients because we really want to learn and work with patients to learn as much as we can about their tumor, as well as help future patients through research studies. And so consenting is really research, but it often can help with clinical trials and things like this later on down the line. And so that's a really critical step to do the most we can and learn the most we can from patient samples. Surgeons then are a critical component as far as removing the tumor and treating patients, and we work in pathology to try to coordinate with them in order to generate and capture the tissue for diagnosis and properly handle it. And what you see is that there's often many different avenues or fanning out of ways that tumors are studied, both for clinical care and for research. And so paraffin or FFPE tissue is something that's generated, and that's kind of the cornerstone of that H&E and microscopic diagnosis. And that's routinely done for essentially every patient. Frozen tissue is really important in today's age. In the past, it might not have been important for clinical care, but there's an increasing awareness that many trials, many studies do need to use frozen tissue. And so oftentimes centers are attempting to freeze samples, and this is really important for patients as well, and to store them in case there's a need for them with a specialized technology. The next step is fresh allocation, and this is a step whereby you see below in the box, fresh tissue can be used to generate living tissue bank or patient-derived models, and these are avatars of a patient's tumor. We're not going to talk about this too much today because it's mainly research, but it's a very exciting new area whereby fresh tissue is kept alive in either cultures or in mice, in order to try and have another portion of your tumor that can be studied outside of your body and do so quickly. And so all of these can feed into DNA, RNA, protein analyses. And on the right, we have a lot of tools we'll talk about today of the different tests that specifically might be applied to do this. And so all of this information is quite a bit of work and quite a bit of data that's generated. And so right now the task is kind of integrating and making all this data useful. So if we go back to the first stage of the process, when that tumor comes out, how is the pathologist going to decide what the diagnosis is? Well, and using any of these tools, how is that going to, what are the rules by which the pathologist might do this? And so what I'm showing here is the WHO, or World Health Organization, classification of tumors. And this is a textbook which every neuropathologist use. And it's basically rules that we follow to try and standardize across hospitals a consensus approach to classification so that the diagnosis will be similar going from institution to institution. And this book is mainly focused on the microscopic or histology diagnosis, but it does discuss and is starting to evolve into something that will incorporate molecular diagnosis as well. And really it is a world diagnosis approach, and this is something that has really improved brain tumor diagnostics since its inception. And the last update to this was in 2007. It's really a large group of pathologists and scientists and oncologists. This last edition had 73 different experts and authors creating it, so it really does represent a groupthink of what the most important things to look for in tumor diagnostics would be. It is a big challenge. If you look at the book, what you'll notice is that there are more than 128 brain tumor types discussed in this book, and I've just listed them here from the book. And so it really, there's new types of entities added all the time, and so this number gets bigger, not smaller. And so it really is a task to try and come up with diagnosis and treatment for this many tumors, all located just within the brain. This is not really counting metastatic tumors of various types that might come from other parts of the body. So what really is driving the incorporation or addition of molecular testing to the histologic diagnosis? Well, technology is really a key driver of change in pathology and diagnostics, just like it is in other aspects of our lives. And it moves fairly rapidly. So the Human Genome Project began in 1990. From around 2003 was when these projects were ongoing and the genomes were first sequenced. And RNA expression profiling, 1992, not too long ago, was first invented. And so whole genome approaches to cancer genetics and genomics came along, and personalized genomics. These were around the 2000s that people were thinking about this. And theoretical level of data now that can be generated for each patient using all these kinds of tools for personalization or precision medicine is really staggering right now and quite unprecedented. So it's been moving fairly quickly. And really, why has it been moving quickly as well? Well, it's because the clinical care of brain tumor patients is changing, and cancer in general. Patients are increasingly responding to therapy. At least the agents that we're using are starting to have impacts on patients' outcomes. And the greater implications therefore are implied in the diagnosis, class, and grade that we give it. A good example is the difference between outcomes and treatments that would be planned for oligodendroglioma versus a glioblastoma. Those are very different strategies and approaches and conversations that would happen with patients with those two different tumors. And the correlations and personalized medicine that are now needed from the data to try to predict how patients will respond to treatments has really escalated. New trials and drugs, as you probably are familiar, are very commonplace. There are many more drug companies and many more drugs than there used to be. And the drugs generally in today's world are designed to act against specific molecular targets. And so the molecular diagnosis is something that guides who might get these drugs and treatments because it's the way in which we identify what patients might respond. Increased pathologist and clinician responsibility is important for designing, evaluating, and supporting these trials and their efficacy. And the complexity really has required much more of a level of teamwork around patient care than was previously required, where each person had their task but maybe didn't have as much interdependency as before. So this all drives change for a positive, better outcome. And I just wanted to mention that what we're talking about today is super ultra topical. Some of you may have heard the State of the Union speech from the President, which was a few weeks ago. And there was just an announcement on Friday, and an invitation went out to many physicians and heads of cancer centers and other patients, and that the President has prioritized molecular testing, which often is what people mean by precision medicine or personalized medicine. Those are essentially the same thing. And all of this just happened. And so really cancer precision medicine it's a big initiative. This is a big part of the initiative and the idea, because cancer precision medicine and personalized medicine is really where many of the advances have been very successful. So these are a big priority as stated by the government right now. And in fact, at the Friday's seminar and announcement, there actually was a brain tumor patient even invited to the White House for this announcement. So it's really quite a topical thing right now in the news. And so one thing that we frequently get asked is, what is the estimated cost of molecular diagnosis? This isn't maybe the most important thing to focus on, but it is good to know where it sits within the grand scheme of a patient's treatment and care. And I've listed here some very rough, very ballpark type numbers just to give you an order of the magnitude of what cost might be billed or might be charged in order to perform a test. And doing a lot of research on this, I can say that these charges are somewhat in line with what we would even pay in a research laboratory setting. And so you can see that H&E is probably the cheapest thing that can happen, around $100 maybe. IHC is similar. FISH and the genomic tests of any type require a lot of work and a lot of labor and a lot of expertise, and so any of those are up into the thousands. And I think, we're going to talk about many different tests today, but they're in similar ranges, generally under anywhere from $5,000 to $1,500. Whole genome sequencing, people hear a lot about this. This is generally still in the cost range of around $10,000 for it to be useful information, and this is generally out of the scope of clinical care at this time and point for brain tumor analysis. And I've just given a reference. An MRI might run something anywhere from $2,000 to $4,000 probably for head imaging of the brain. It would be at the higher end and might even cost more with specialized techniques. So the testing that you get, which is usually done once for a brain tumor patient, is costly, but it's actually in the relative picture of overall patient's care, not that costly, particularly in that it's not going to happen over and over again like an MRI type of event. And I think that's really important. So it's, and people and patients do get involved with the cost depending on where their testing is done. The tools for molecular testing, we're just going to go through those. And right now, the first one we're going to start with is the immunohistochemistry, which is looking at the protein biomarkers in patient samples. Immunohistochemistry really is one of the first molecular tests that was developed, and really is generally available at all pathology labs. So this is something that's available to all patients, generally speaking. It looks at, basically it's looking at one protein, which is a product of maybe one gene, and it's one test. And so it requires usually just one slide from a section from a patient's sample in order to perform the test, and can be done on even the smallest biopsies. So it's very widely accessible, and it's fast. It usually takes most labs 1 or 2 days to perform this type of test and get the results back to the pathologist, who can then incorporate it into their report, which may take a little longer, but is generally very rapid. And glioblastoma is a useful model for thinking about molecular immunohistochemical diagnosis and how that's been changing over time. GBM is our most common adult primary brain tumor. As many of you may know, it's resistant to chemo and radiation for the most part, although you can have some responses. And really the question there is some patients live longer than others or respond better than others. And can any of these types of tests help us to identify who that might be and give them more accurate information on their tumor? And so immunohistochemistry used in diagnosis, again, looking at the protein expression, or such as a proteomic type test it would fall into. Really, there's a couple of markers that I wanted to talk about that are the most common or most useful right now. And those two are OLIG2 and GFAP. And both of these are useful, for instance, as glioma markers. And so if you have a pathologist who's approaching a tumor and feels that it's a glioma, they can run an immunohistochemical test for one of these markers. I've shown OLIG2 here. And what you see is that in the brown, these are each one cell nucleus staining for OLIG2. And having OLIG2 sort of indicates that the tumor is most likely in that glioma category. So it helps to narrow down and really helps to tell us what the diagnosis or that first step in an approach to a patient tumor might be. And GFAP does a similar type of thing. The immunohistochemistry can be used also to look at mutations. So the pathologist may use it to look at IDH1, which is a very interesting and common mutation in a subset of patients with gliomas, both adults and children. And we have tools that will see a specific point mutation, a change in the DNA, which leads to a change in the protein. And the one that's most commonly used is an antibody, IDH1, R132H is the name. And really this helps us to identify subsets of patients who have an oligodendroglioma or an astrocytoma that is not a glioblastoma type grade. And there's an example shown below. You can see all the brown are the nuclei or tumor cells that are staining strongly with the IDH1 mutation. So the brown indicates the patient has the mutation. You can see over here a neuron that's a normal neuron, which was present also within the biopsy material that was removed. And the normal neuron does not have the mutation. And that indicates that the stain is working well. We have several of these mutation-specific antibodies. Another common one is BRAF, V600E, which is felt to be a driver gene, very important for diagnosis and potentially for treatment of things like pleomorphic xanth of astrocytoma or PXA and ganglioglioma. And there's a number of others such as ATRX, TP53, which can be used to sort of evaluate tumors and mutation status within those tumors. And really these are things that, if we think about it, are things that we feel through their mutation are actually helping to drive or make the tumor grow. So very important to evaluate those in a patient's sample. Another thing that immunohistochemistry can do is help us with determination of prognosis. So this molecular test, Prognosis, and some of the tests we're going to talk about here, would tell you maybe an estimate of how fast is my tumor growing, or where in the spectrum of other patients with this diagnosis might my tumor lie as far as its aggressiveness. The one that's very commonly used that people might see in reports is the MIB1 or Ki67 proliferation index. That's shown below here. Again, the brown nuclei are cells that are tumor cells that are growing within the tumor, versus the blue circles are tumor cells that are not growing. They're just sitting there. They're not any longer contributing to the tumor growth. And by calculating how many of these cells there are growing within a tumor, sometimes this gives us a very helpful estimate as to how aggressive the tumor might be. And again, going back to prognosis, the IDH1 R132H mutation is a very important diagnostic as well as prognostic marker. IDH1 R132H, the presence of that mutation generally marks patients who have better prognosis and helps to distinguish those from patients who may have a slightly or less favorable prognosis. All can be done on one slide with immunohistochemical tests. Often patients are considering clinical trials of experimental agents or novel therapies that are directed against some of these drug targets or drivers of tumor growth. EGFR, epidermal growth factor receptor V3, is one such target, as well as the tumor suppressor P10. And in a paper by Ingo Mellinghoff in the New England Journal, these two genes and mutations were felt to be interacting in a way that would help us diagnose or predict who might be a better responder versus a worse responder to EGFR drugs. And so the presence of EGFR V3 and P10 protein by immunohistochemistry was one way that immunohistochemistry was used to help try and identify patients. Now there's a lot of practice to doing this. And many of these studies, in research trials are being examined, and currently we don't use this in clinical practice quite as much. So I list it as an example, but right now it's still being evaluated how this would help to indicate whether patients would respond or not. And I think right now it's in larger studies and trying to look at different drugs. Another one that people may have heard of is Celldex, is a company that has a trial, for instance, where EGFR V3 is used as a vaccine. And so patients are stimulated to try and have a response to EGFR V3 in their tumor using their own immune systems. And this is another place where patients considering that trial, if they have an immunohistochemical test that tells them they have the V3 mutation, then they might consider that trial more seriously than if they didn't have it and wouldn't be eligible. Immunohistochemistry has been generally done on just single types of proteins or targets and with single antibodies and single tests. There is some attempt to expand that to look at what we call multiplex testing for immunohistochemistry, or different types of expression of proteins and RNAs that might lead to proteins. One is phosphoantibodies and multiplex phosphoantibody arrays, and trying to look at activation states of different pathways. And so this right now is research and not really clinical at this time. You may have seen places that offer access to such testing, but really for most academic centers it's considered still research testing, not proven to be useful for clinical care. Expression profiling and the Cancer Genome Atlas subtypes is something that in research realms we use very extensively to try and understand tumors better, looking at their RNA by different genomic technologies. However, RNA is not yet clinically used very frequently for GBM patients. There are some tests out there for expression for GBM as well as breast cancer, and these are some of the names of companies that you might have seen offering such a test. But again, mostly researchers consider these too early still for implications of brain tumor patients, and most hospitals don't run them. Although these types of tests are medically available or clinically available. So tools for personalized medicine, the next step is that we'll talk about the tool of cytogenetics, which looks at DNA and changes in your DNA. And generally a lot of the tests for cytogenetics are looking to measure the number of copies of a gene. Is there a gain or loss, too many or too few copies of a gene that's important for the cancer? Does the gene appear intact, or did it get rearranged or mixed up in some type of rearrangement of a patient's DNA within the tumor? Or is a gene fused to another gene, making some type of protein that causes tumor to grow that never is seen in a normal cell? And the best example of use of cytogenetics in patient care is probably oligodendrogliomas, which are generally grade 2 or 3 tumors. These are fairly rare tumors and occur in the brain with a very striking pattern of halos or circular cells with spaces around them shown down here. This type of histology is sometimes very variable, so looking at it through the microscope, this can sometimes be hard to diagnose, and you might not see the features needed. And so a number of years ago, it was noted that a genetic event, rearrangement of genes and fusion of DNA of chromosome 1 and 19, was very reliably seen in at least 80% or maybe even more of these patients, leading to loss of chromosome 1 and 19 in oligodendroglioma patients, and that this was basically very diagnostic association that was helpful. And it's fairly important in that oligodendroglioma is one of those tumors that has IDH1 mutations as well, very frequently, and so has an improved prognosis compared to some other tumors and can respond to treatments quite well in many patients, so it's important to identify it. The genetics of oligodendroglioma, we just talked about the translocation or the co-deletion of 1p and 19q is the most important aspect of that. But what I just wanted to show was that in the research realm, there have been many different associated genetics events that are known to drive progression to higher grade or less favorable prognosis. And in the past, we didn't have ways to check all of these realistically for patients until we developed some molecular type testing that is able to do multiplexing or able to check multiple genes and events at multiple times. And so you can see there's a lot of data and information which one test might not be able to capture. The standard test that we've used for many years for diagnosis of brain tumor patients is the 1p19q co-deletion FISH test. So cytogeneticists use this in order to give diagnostic and prognostic information. And it's shown down here in an example where basically each cell is looked at its nucleus to see how many copies of the 1p and 19q regions there are. Here's a nucleus that has green and red, and 2 pairs is normal. And here you're missing, say, the green signal, and so that's been deleted. And so that cell has lost the signal for lost DNA in that region. 19q also here is lost. The red signal is lost in this instance as well. So this patient would be a co-deleted patient. The presence of co-deletion essentially rules out glioblastoma, so it really helps a pathologist to pick between hard cases of oligodendroglioma versus glioblastoma. And again, these patients, it really does matter. Glioblastoma, this diagnosis is generally something that's quite different. So in this case, for instance, we had a patient who was diagnosed with a glioblastoma at one center, and then by doing FISH testing later was found to have what was more likely an oligodendroglioma. And this made more sense because the patient did have a good long-term response to standard chemotherapy and radiation, and that it would have been exceptional for a GBM maybe to have that, whereas in this case for an oligo it was more understandable and expected. So it really can help to change the diagnosis. What about astrocytomas, glioblastomas, some of the more common tumors? They also have multiple genetic events, each of which tell us a great deal of information about the tumor. Even just something like a primary glioblastoma, the most common variant, has 4 or more different genetic events or molecular events that need to be tested for and are very important. So the diagnostic tools that we use for that is amplification of EGFR is a good example of something that helps us diagnose, like the 1p19q GBMs. And to test for this, in the past we've used tests such as FISH or SISH, which are single tests, again, to look for how many copies of the gene we have. EGFR, when it's amplified or has too many copies, almost has hundreds or greater than 10 for sure copies. And about half of patients, or 40%, have this in GBM, and it really is helpful in diagnosing GBM and confirming that. This is a picture of that. So there's a picture here where we've taken samples from different regions of the brain around a brain tumor shown here. And you can see each little brown dot is a copy of that EGFR gene present within a cell. And here's a cell way far away in C from where the main tumor is in B here. And you can see that even though there's a normal cell here right next to it is a cell that has too many copies of the gene, and this diagnosed even on a single cell that it's a tumor cell. Same goes for this region away from the tumor here. So it's very powerful for identifying tumor cells, but it's just one test again and one gene. So can we use a different test to try and capture all those different genetic and molecular events all at once? Well, luckily we can. So recently people have started using what's called ACGH, or Array CGH, to help multiplex and do cytogenetics with with much more efficiency. And in brain tumors, you think about how many different targets we have or molecular events, and this is really too many FISH tests if we want to know all of these different events in the same patient. So the array-CGH really is what we call a whole genome test, and it checks all the regions of your chromosomes and DNA. And how does it do this? Well, it really is like a FISH test that's similar to a FISH test in that we take DNA from the patient sample and DNA from a reference, and we allow them to compete. And instead of just applying them to one glass slide, we apply them to a glass slide that has multiple pieces of DNA. And so the two normal and the patient's DNA from the tumor try to compete on the slide, and the competition, whichever sticks most to the different regions of the chromosome, that shows up as a red or green signal. And this is a slide with the green and red kind of diagrammed on there, and when they're together they make yellow. And each dot, there's millions of dots on here, each dot is like having a separate FISH test. And so that's really like 1 million FISH tests on one glass slide, and it gives us a whole picture of a patient's DNA and tumor genome. And how can this be used? Well, it's used to replace FISH most commonly right now. And I'm just showing here some examples from pediatric low-grade astrocytomas, which we've studied a lot at our institution. Pilocytic astrocytoma is a very common tumor in kids. Occurs in the cerebellum and has very characteristic histologic appearance, but in many cases it's difficult to tell what the tumor is. And in 2008 it was described by a number of groups that there was a fusion of the BRAF gene with another gene, KIAA1549, and that fusion event or molecular event produced what was a driver oncogene and diagnostic gene event for diagnosing pilocytic astrocytoma. And so recently we've applied Agilent array-CGH to develop a test called OncoCopy, which is a clinical test at our institution and looks at the whole genome and can find BRAF as well as 41 or 42 other genes and aberration events or molecular events all in one patient tumor. And so you can see an example of that here. The red means there's too many copies of that relative to the normal, and each one is like a FISH test again. And the green is too few copies. So if you look, black means it's the right number of copies. So you can see this blip here means there's too many copies involving this BRAF gene. And so these tests are now able to be uniquely designed to target things that are brain tumor specific. It's not offered yet very widely at very many places, and so I think this is something that's very new that's being rolled out at a number of different hospitals. And whole genome, really the nice thing about it is you can go back to the data without having to actually do another biopsy or without having to do any interaction with the patient. If there's a need or interest to go back to the data and look at other portions of the genome besides this list, you can do it very easily in just using analysis methods on the same data set. And so it's really a very nice assay to have. And takes about 2 weeks to do that type of test. There's another example just showing in a pediatric diffuse astrocytoma unique events such as MIBL1 gain or amplification event is also something that can be detected and can replace the need for developing individual FISH tests for each of the tumor types. So the last thing we'll talk about is DNA sequence and mutation detection. This is the newest type of tools that have been used. One thing that people may be familiar with is MGMT testing. This looks at DNA sequence that is a methylation event of DNA in specific sequence regions and comparison of that. It uses a technique which is methylation-specific PCR in order to determine what the methylation or epigenetic state of a tumor sample might be at the MGMT gene. And one key event is it's very useful test and used on almost all high-grade and low-grade gliomas in patients right now. Immunohistochemistry, for instance, in comparison, even though it can give you a little bit of information, doesn't yield reliable or useful information specifically for brain tumor patients at this point in time. So methylation testing is really critical, and that's because it yields prognostic and predictive information about who will respond to standard therapy. And so patients who have a methylated MGMT promoter will survive and have better prognosis and response to treatment than those who have unmethylated. And so it's really a cornerstone of analysis for patients right now. What a lot of excitement is developing is for precision medicine and personalized medicines around multiplex sequencing, so like for the cytogenetics with the array CGH. The more sequence you can generate, the more you can check multiple regions and gene targets that help patients and their diagnosis. And so right now there's many different types of drugs that are directed against particular point mutations and sequences, and that's driving a lot of clinical trials and a lot of activity in this area. And so really the idea is to generate as much information as one can on a tumor. And multiple hospitals have now, and companies have come up with sequencing panels or assays based on next-gen sequencing. One of the most common is Illumina and exome sequencing. And our test at our hospital is called OncoPanel. This is a clinical research offered test for clinical trials using this type of sequencing. And the idea here is we look at around 275 cancer-causing genes. It takes about 4 weeks. It is the same, essentially the same test as what Foundation Medicine runs and many other companies. And it gives you really lots of information about all these genes and the whole gene. If there are any mutations in those genes, they're usually able to be detected as a base change shown here and multiple reads seen from each of the patient's tumors. So right now at our hospital, for instance, you can run many different tests on many different patients in very high throughput. For instance, 150 brain tumor patients were profiled in the first 6 months of our program, which is run here. And really now thousands of profiles are being generated at hospitals around the country. And again, these can be designed, detect, and often do cover many of the brain tumor patient-specific relevant targets. And so if you look, you can have these types of events and reports generated, which have a lot of data now in them. And in fact, this has created a need. And the need is that we need integrative reporting, and this is something that you may see as well, which is when you're generating multiple tests, like we've talked about, whether it be OncoCopy or OncoPanel and sequencing, we need to more integrate these types of reports and generate diagnostic, actionable target information in as holistic a way as possible for each patient and communicate that to the oncologist or treating team. And this is something where pathologists are actively coming up with better and better ways to do this, and is fairly new. One way that this type of integration really matters is in trials. We like to know the exact status of a pathway. Is there a tumor suppressor or some type of activation potential that really is critical for the pathway, and this patient should really consider a trial of a drug more seriously than another drug because of their specific changes in their tumor. This is an example here where we took the OncoCopy result and the OncoMap result, another sequencing assay, and looking just at the patient. Here, this patient with an adult GBM had P10 loss through copy number cytogenetic loss of chromosome 10, and the other copy of P10 that was left behind had a point mutation which inactivated that gene. And so this patient would have really quite a big problem in their P10. They don't have a functioning P10 anymore. And in this case, they would have heavily considered, for instance, a trial of the agent VKM, which is a pan-PI3K inhibitor thought to act in some ways through P10. And so they would be eligible for this trial strictly, whereas there might be no eligibility if they didn't know this type of status, so it can really aid that. And it has other examples of BRAF V600E, for instance, is a very important target as well for pediatric gliomas and rarer adult tumors. And really right now there's a big push at our center and many others in the community to try and come up with next generation clinical trials which use all this information integratively in order to try to more rapidly find new improved treatments for patients. And how are we doing this? Well, really what happens is that you have multiple arms of a single trial whereby a patient gets genomic data, say for GBM, and all of this genomic data then helps decide which of the agents they would pick from, but they would get one treatment while on the trial They would not just be shuffled off to find a trial on their own, and that every patient would be able to go on the trial, but would go on an arm that was customized for them. And so this is really what we're working hard towards right now, so that patients have more options and that research advances much more quickly. And I think I'm just going to end on this slide. A lot of times people want to know, what testing should a tumor get? We can't go through all 128 brain tumors fully, because each one has a customized set of tests that are indicated that the pathologist is picking from. But if we just take a couple examples, such as adult GBM, IDH1 R132H is probably one of the most important tests to run for an adult GBM patient, whether it be by sequencing or immunohistochemistry, MGMT methylation PCR, and then some type of assay, whether it be a multiplexed oncocopy or oncopanel type assay for sequence and cytogenetics analysis, copy number analysis, or it can be focal events where you look at just by FISH or sequencing for just one gene. Either way, those are very important testing modalities for adult GBM. Pediatrics and their gliomas, for instance, BRAF V600E, BRAF duplication by some type of test, IDH, and broader testing for oncocopy or oncopanel are, for instance, approaches that we would use at our hospital for every single patient, should there be enough tissue. And that gets to my last point, which is really there's a lot more need for patients and clinicians to be understanding that tissue quantity is a major consideration. All of these tests require certain amounts of tissue, and it's more than we used to use for routine diagnosis. So molecular diagnosis, each test uses 10 to 20 slides on average, and clinical trials also require tissue for their analyses and for patients to go on trials. So right now we have some patients and some situations where we have to be really working together as a team with the patient and the treatment physicians to decide what are the most highest priorities and how do we want patient tissue used. And so that's something that really patients should be aware of going forward, is that they are part of that conversation. And so I just want to acknowledge the ABTA for this wonderful format of educational seminars and webinars, and thanks to their program in particular for their support of young scientists in laboratories across the country, where they've done a great job supporting generation of all the data led to these molecular tests. And I'll stop there and answer any questions.
Thank you very much, Dr. Ligon. That was a very informative presentation. We've received a number of great questions, so without further ado, we'll ask the first one. Can every doctor administer precision medicine?
I'm sorry, can you say that again?
We had a question whether every doctor is able to administer precision medicine.
So precision medicine is a term that encompasses a lot of spectrum of what we just talked about. Every doctor is able to do precision medicine in the sense that, as I said, immunohistochemistry tests are available at almost every hospital. And so sort of having pathologists use and generate data on that and incorporate it and talk about it with the oncologist and patient is something that can be done everywhere. The spectrum of testing that we just described is particularly those multiplex tests. Those are fairly new, and so those have not quite entered into every single hospital's regimen of how they approach patient care. Certainly all the major cancer centers have major efforts for those types of tests, and so you know, it really does depend. It's a very good question. It does depend on where you're being seen. I think for brain tumor patients, given their particular needs, we do encourage trying to go to centers that would have availability of these types of tests from the very beginning, because the amount of tissue and things available, all the knowledge needed to make these tests happen is all coordinated, and so it's important to get to the right hospital at the right time in your care so that you have options for these types of things.
Thank you. Our next question seems to have two parts. Is molecular testing part of the standard discussion between doctors and patients? And what can patients do if a doctor doesn't bring up molecular testing?
Patients should bring up molecular testing for sure with their physicians and should not be shy about it. I think that is a really important feature. The patients have a role. The physicians may or may not be as aware of the modern practices that are going on at various centers. So I think both questions are kind of indicating the understanding that there may be a wide variation from center to center, and that is the case. And so I think it really is important to bring that up. Many physicians or treating physicians would be maybe able to offer consultation where you send pathology or send it for analysis to another site, and certainly ours, as almost any other cancer center, we have lots of patients who send their material for analysis and molecular precision medicine analysis so that their physician can have access to that too if they don't have it in their local pathology group. So, but I think it is something that basically patients should ask questions about. And as, for instance, the test for IDH immunohistochemistry is a very simple test. And so it's a very important test for each patient, for instance, with a glioma, and really is not not something that's out of the reach of most centers to perform.
So would this be blood or tissue that is used?
Almost all of the tests that I talked about today, those all only require your tissue or tumor tissue for analysis. Some of the sequencing and multiplex tests are sometimes run clinically with patient's normal blood or a normal tissue sample from a patient as a comparison. But for the most part, everything is able to be run, everything I described today is able to be run on paraffin tissue, which is very important because if you have a tumor surgery somewhere where they were not able to do frozen, which is the most places the case, paraffin tissue, we've worked hard to develop all of these assays so that they can be applied to routine paraffin tissues. But it does require a certain amount of tumor to do it, and occasionally patients do not have enough tumor in order to run all the tumor tests. But usually some of the molecular tests can be run, just not all of them.
Is there anything a patient can do if they've already had the tumor biopsied and testing was not performed, or there's not enough tissue for all of the testing?
Yeah, so if the first step is to basically discuss with your physician and have a conversation with them about what else could be done to generate more information or data. And the usual answer to that is that yes, there usually is something more that can be done, but it often might require submitting or coordinating submission of your sample to another hospital or cancer center that has those tools available. And again, that's standard practice, and every single cancer center or tertiary care center does accept consultations and can work to generate more molecular data than maybe was initially done. The tissue is preserved, it's not destroyed generally, and so it is able to be done at other sites if the patient or a clinician asks for it.
Thank you. Now we've just got time for a couple more questions, and these are a little more generalized. What is the difference between molecular testing and genomic testing?
They are one and the same. So the number of terms that you will hear out there are genomics, genomic testing, molecular medicine, molecular tests, molecular pathology, precision medicine, personalized medicine. All of these things are very general terms which usually are talking about sequencing or copy number analysis or immunohistochemical analysis for specific mutations and proteins. So everything we talked about today would fit under that. Because of sort of just language, often people are talking about sequencing or copy number, more sophisticated multiplex arrays, which are looking at multiple genes and proteins instead of just one at a time. So often that's what people are talking about, but really all those terms are used and mean the same, essentially the same thing. That's a very good question.
Thank you. And our final question is it possible for a lower grade tumor to suddenly turn into a GBM, and does that require additional testing once it's become more advanced?
Yes, so for low-grade tumors, many low-grade tumors, it really just depends on the exact diagnosis and class, but there are a number of low-grade tumors that have a certain rate of transformation into a higher grade tumor, and so one of them, lower grade gliomas, can over time become more aggressive and turn into something potentially a glioblastoma. So that's definitely a pattern that can happen. One of the things that happens is when different pathologists and different genetic and molecular testing data is applied to a tumor, sometimes tumors that were originally called low grade, with more tests, we may be able to determine that even though we had hoped that originally it was low grade, sometimes the genetics and molecular change the diagnosis or the grade into a higher grade tumor. Sometimes they change a high grade diagnosis into a low grade tumor. So really they are there, and they are, it doesn't happen super frequently that you change diagnosis, but it happens enough that it's very much worth having second opinions and consultations, given that a patient doesn't have to travel, only their tumor tissue sample has to travel. It's a very important thing to do for a brain tumor diagnosis, to get second opinions from places that can run those types of molecular tests.
That's all that we have time for today. I want to thank you all for joining us, and thanks once again to Dr. Ligon. For more information on the topics discussed here today, or for more information on brain tumors and their treatment options, our licensed healthcare professionals can provide you with support or help you navigate information available on our website. Please feel free to call the ABTA Care Line at 1-800-886-2282. Let's pause for a moment to conclude our webinar recording.